Hi students, in the very first video we have gone through the general characters of pteridophytes as well as outline classification as per spawn. In second video we have learned about morphological and anatomical characteristics of Selaginella. In the third video we will learn about reproduction in Selaginella. So let us begin with the reproduction of Selaginella. The sporophyte of Selaginella reproduces by vegetative and sexual methods. Vegetative methods include the germination or development of new young ones by means of tubers. So, through the tubers, vegetative reproduction taking place in Selaginella chrysocolis. By the bulbils, vegetative reproduction is taking place in chrysorhizos, and by the fragmentation of the plant body, it is taking place in the species Selaginella rupestris. Bulbils are actually dormant buds which are produced in aerial branches, while the tubers may be aerial or underground parts of the plant. In Selaginella, reproduction is also taking place by the spores. Numerous haploid spores are produced in the sporangium. The sporangia are located in the sporophylls, and sporophylls are compactly arranged to form a cone or a strobilous. All the species of Selaginella form strobilous or cone at its apices, as we have seen that in second video. Generally, the strobilus occurs terminally on side branches. Selaginella is heterosporous because it produces two different types of spores, and therefore sporangia are of two types, that is microsporangia and megasporangia, that you can see. Microsporangia are male reproductive structures and megasporangia are female reproductive structures. The sporophylls associated with two types of sporangia are designated as microsporophylls and megasporophylls respectively. So, within the microsporophyll, microspores will be there, and within the megasporophylls, megaspores will be there. In this slide, you can see two different kinds of strobili. In the first kind, here in the upper zone, you can see microsporophylls, and in the lower portion you can see the microsporophylls. Whereas in this kind, one side is representing microsporophylls and another side it is representing megasporophylls, and this megasporophyll is enlarged here and shown in detail, whereas this microsporophyll structure is enlarged here and it is showed in detail. So, if you look here, is present stalk, here is present stalk, here you will see a ligule, here you will see a ligule, and this structure is microsporangium and this bigger structure is megasporangium. Inside megasporangium you can see 3 to 4 large megaspores, whereas here inside the microsporangium you can see numerous small sized microspores. So, this kind of structure is generally seen in the cone of Selaginella. There is variation in the distribution of sporangia as we have seen in the previous slide. where within the strobilus either one side is with microspores and another side is with megaspores. Another possibility is there that upper part will be related with, or it is representing microspores, whereas the lower part is representing microspores, and here it is mega. So variations can be seen. Let's see the structure of sporangium. The mature sporangia are stalked with two-layered jacket. The microsporangia are slightly elongated and reddish to bright orange in color. Megasporangia are larger in size, as you have seen in the previous slide, and microsporangia are actually small in size and frequently they are lobed. The megasporangia are whitish yellow or light orange in color. The spore grows and develop the gametophyte. The haploid spores germinate to form endosporic gametophyte. Let us understand what endosporic gametophyte is there. If this is a spore and if gametophyte is developing within the spore, it is not coming out of the spore. So the entire development is taking place inside only. This is called as endosporic, and this kind of structure is seen in Selaginella. But in some other pteridophytes, this is a spore and this spore germinate outside and gets enlarged. So when this big structure is formed, this is actually called as exosporic. But in Selaginella, it is taking place inside the spore only, like this, and this is endosporic. So the development of microspores and megaspores generally starts while they are still inside the respective sporangia. So actually when it is present inside the strobilus, like here microspores and here the megaspores, the development has started when it is present inside the cone. Therefore, the spores are shed at multicellular stage. Male gametophytic structure we can see now. The microspores germinate to form male gametophyte. Here the developmental stages can be seen. So here you can see the jacket cells. Here you can see the jacket cell increases in number. Here you can see there is formation of androgonial cells. Then they divide further and there is development of antheridian cells, and this development goes further, and from unicellular structure there is formation of multicellular structure. Then there is formation of anthrozooid, and this anthrozooid liberates out this structure you can see, and it has two flagella, as you have seen in the general characteristics that it is biflagellated. So this anthrozooid is biflagellated. So this way from single cell multicellular structure is formed, and from that individual numerous biflagellated anthrozooids are formed. Now we will have a look over female gametophyte. Like male gametophyte, the megagametophyte, that is female gametophyte, development of Selaginella begins while the megaspores are still inside the megasporangium. Archegonia originated in the apical region of the female gametophyte. Each archegonium develops from single superficial cell, that is called as a archegonial initial, which divides periclinally to form primary cover cell and central cell. The details we will see on the next slide. Here you can see this is a megaspore, and as I said you that this is endosporic, means the development is taking place inside the spore. So this is actually the spore, and inside that there is a beginning of development. So there is formation of vacuole, and here you can see free nuclear divisions are taking place, and the same structure here you can see in enlarged structure. Here numerous free nuclei can be seen, and this is vacuole. Then in the next stage, here you can see that lateral side of the free nuclei there is formation of archegonia here, and in the peripheral zone there is formation of rhizoids. These rhizoids are for absorption, and here somewhere you can see there is development of archegonia, and this small portion is enlarged here, and here actually there is archegonium represented. Now we'll have a look on fertilization. In majority of the species, fertilization takes place after megasporangium gets settled on the substratum, but in some cases like Selaginella rupestris, fertilization occurs while the female gametophyte is still within the sporangium. Biflagellate sperm, which is haploid in nature, are liberating, and then they swim to the archegonia through a thin film of water and fertilize an egg, which is also haploid. So haploid egg and a structure that is biflagellate sperm, or you can call antherozoid, they fuse together and there is formation of zygote. So as you can see that this is a haploid structure, and this is also a haploid structure. This N and this N together forms 2N, that is diploid zygote. Here let's see about life cycle pattern of Selaginella. So let us start with the sporophyte of Selaginella. You can see here, this is the plant. Here you will see the leaves, rhizoids, rhizophore and roots. Okay, and here you will see the strobilous. So this strobilous is represented here. So those which are large structures, these are megasporophylls, and some small structures are also there. These are microsporophylls. As you know that these are diploid structures. So these undergoes meiosis, and meiosis divides that into half. So after meiosis there is represented megaspores, which are N, and such small structures they are represented. These are also N haploid structures, microspores here and megaspores here. So individual microspore haploid is represented here, and individual megaspore, which is also haploid, that is represented here. So inside this you can see male reproductive structure, and inside this you can see archegonium. So this male reproductive structure is individually represented here, that is in the form of antherozoids, and here individual reproductive structure is represented here, that is archegonium. So these anthrozoid enter inside and thus they fuse together. There is egg which is N and the spermatozoid which is N. They undergoes fertilization and there is a formation of zygote which is 2N because Thecan and Thecan fuse together and that is represented here and that is called as zygote. So this divides and redivides and grows further and that is called as young sporophyte which is attached to the female gametophyte. So female gametophyte is still there and there is young sporophyte. When it grows further, it detaches from the gametophyte and this portion individually grows into new sporophyte. So this way it grows in a cyclic way and this is what we called as life cycle. If you see, there are two parts. If you see this part which is sporophytic generation and it is diploid, and here you can see gametophytic generation which is haploid. So this is what a life cycle of Selaginella. Thank you.